All right, welcome back, microbiology. Today we're going to be talking about immunity. We'll divide this into two parts. Part one will be non-specific defenses of the host, and part two will be specific defenses. So for our host defenses, some terms that we'll use the term resistance, which means the ability to ward off disease through body defenses, and susceptibility would be how susceptible are we to that disease, or how vulnerable are we to a disease. When we talk about non-specific resistance, we're talking about just a general protection that would protect against anything, not anything necessarily specific. So we have two lines of defenses for that. For our non-specific resistance, our first line is going to be what a pathogen would encounter first, which is going to be the skin and the mucous membranes. Our second line of defenses would be what they would encounter if they are able to get through that, which would be phagocytes, inflammation, fever, and then antimicrobial substances. If they're able to withstand all of that, then they will encounter, and these are going to happen somewhat overlapping here. You would have our specific resistance, which would be targeted towards a specific pathogen. So we call that the third line of defense, and that's going to involve antibodies that we make to try to target, clump together, neutralize, and destroy these different pathogens that come into our bodies. So innate resistance would be the resistance that we're born with, and that's going to be the first and second lines of defenses. So of course our immune system is going to be important, and for this, just some structures that we'll see involved. We've got our tonsils. Those are going to play a role in trapping pathogens as soon as they come in through the mouth. We have our lymphatic vesicles, our lymphatic circulation, and that's going to go through the lymph nodes, and that's going to help pull fluid and pathogens from tissues in our body. Our spleen, the thymus, our bone marrow, we're going to have our erythrocytes produced, and then our lymphatic vessels. So some mechanical factors. This would be for our first line of defense. We're going to have our skin and our mucous membranes. So we'll start with the skin. For the skin, this is our surface barrier, and as you've learned in Anatomy 1, it has these two major regions. That would be the dermis, which is the inner, thicker portion of connective tissue, and then the epidermis, which is that outer, thinner portion of epithelial tissue. As you move into the dermis and epidermis coming above, like superficial, to the muscle and fat below, you'll notice all these properties with the skin. So one of the best things that our skin does to prevent pathogens from infecting us through our skin is that the cells are layered continuously, one on top of another, packed really tightly, and that prevents them from just crawling right into our circulatory system, for example. And because they produce these layers of keratinized cells, especially on the edges where they literally die and become waterproof, and then they slough off our bodies, that waterproof layer is really good at preventing microbes from getting into our bodies. So for the mucous membranes, these consist of epithelial and connective tissue layers. They line the gastrointestinal tract, respiratory, and genitourinary tracts. They inhibit the entrance of microbes, but they are less protective than the skin, because you got to think in these areas where you have mucus, like in our lungs, for example, it's not far from our vascular tissue. It's right above it. So it's a little bit closer to, our circulatory system, for example. So they secrete mucus, and that to prevent these tracts from drying out, and that mucus can trap microbes, and then phagocytes can phagocytize those microbes. They contain cilia, so it's important to continue to secrete mucus and then to get that mucus moving so it doesn't stay in one place where those microbes might be able to continue to grow. So some microbes actually do really well in the presence of mucus, in which case all the more reason to keep it moving. For our lacrimal apparatus, that's where we're going to produce tears, and those are going to flow over our eyes, and the produced fluid helps physically flush the microbes out of the eyes, And then the lysozyme that's in that fluid, in those tears, can help break down the bacterial cell walls to also destroy the bacteria at the same time. So we flush and destroy through our tears in the lacrimal apparatus. For our saliva, so produced into our mouth, what that's going to do is it's going to dilute what's in our mouths, dilute the microbes if they're condensed in one point, in one little glob, and it moves them away from the teeth, which protects our teeth, and it does contain several, actually, antimicrobial compounds. We mentioned lysozymes. That's also in our tears, and that breaks down those bacterial cell walls. So you're also going to have that here. Now, your lysozyme gets depleted after meals. So it's prepared for mealtime. When you eat, it's released, and it helps fight bacteria that also cause cavities. But in between meals, it has to be replenished. And if you continue to eat sugary foods, then you're not going to have enough lysozyme to break down those bacteria that will feed on that sugar, in which case you're more likely to get cavities. So they say if you're going to eat sugary foods, eat them with meals and follow them by something that is more alkaline, some kind of basic vegetable at the end of the meal, rather than trying to snack on sweet things in between meals. So something else that our saliva produces is something called secretory immunoglobulin A, abbreviated IgA, and we'll look at these different types of immunoglobulins or antibodies at the end. But this one neutralizes pathogens in the oral cavity by helping them basically neutralize their effects. So our epiglottis, this covers the opening to the trachea, and this keeps microbes out of the lungs when we swallow food. And again, that's that leaf-shaped flap of cartilage, and it's located in the back of the throat. You see it here depicted in the diagrams. So just behind the tongue, in front of the larynx or the voice box, and its primary function is to act as a switch. So when you're swallowing, it directs food and liquids into the esophagus, what we call the swallowing tube, and it prevents them from entering the windpipe and the lungs. So when a person is not swallowing, the epiglottis would remain upright, allowing air to pass freely into the larynx and the lungs. So during swallowing, that epiglottis folds back and it covers the entrance of the larynx. And then it returns to its upright position afterward. So that's a really important structure in our bodies to keep foods going into the stomach when we swallow. And that's really important because if there are microbes, when they get into the stomach, that low pH hydrochloric acid mixture is going to be able to destroy most of them that you're not going to see in your lungs. So some other mechanical things that we can do, one would be urine. And that's going to be for males and females for the urinary tract, but for males, it's also going to be flushing through the urethra to keep microbes from reproducing there. So the urine flushes the microbes out of the urethra. And then for females, because they have separate tubes there, access points for the bladder and the vagina, they also have vaginal secretions that flush microbes out of the vagina. So some chemical factors that we'll see on our bodies. One would be sebum. So this is an oil that prevents drying, and it provides this protective film over the skin. We also have perspiration. When we work out, we get hot, and that sweat also flushes microbes off, and it contains lysozymes. Auto spell check there. In our stomachs, we have gastric juice. We talked about that hydrochloric acid, that low pH. It's very hard for microbes to live in that environment. And then we have the vaginal secretions, the urine, and then, of course, in our blood, we have a few interesting materials. We have transferrins, and what those do is they actually bind iron, and that keeps the bacteria from being able to get it. And bacteria need iron to survive. So if all the iron is bound up in these transferrins, then the bacteria don't have access to it. There's also some protein complements, and what they do is they attract phagocytes to the microbes, and it helps puncture their membranes as well. So it can help destroy any microbes that are floating around in the blood. And then there's a picture of that sebum on some oil, and it just prevents drying and provides an extra layer of protection over the skin to protect it from microbes. So something else that we have that helps prevent infection is our normal flora. So the normal microbiota, these assist in preventing the overgrowth of pathogens through several mechanisms. Number one, because they're already there, they're already fighting for the food that's going to come into that environment. So if a pathogen tries to come in, it's got to fight for food against all these other microorganisms that are already adapted to the environment and are doing very well. So that's going to make it difficult for them to compete for food against them. Also, those normal microbiota, they're going to produce different chemicals that allow them to dominate that environment. So that's going to be harmful to any other organisms that try to move in and start taking their food. And then altering conditions will affect their survival, such as pH and oxygen, that these normal microbiota can influence. So for phagocytosis, this would be the ingestion of a microbe or a particle by a cell. So part of the body's second line of defenses. The white blood cells engulf and destroy the bacteria. We'll talk about some different white blood cells, and this occurs in 4 stages. So our first white blood cell we're going to have is something called a neutrophil. You'll see what its multi-lobed nuclei looks like right here. This is the most abundant and is the first to the scene. However, they do die quickly. Macrophage, you hear that one come up probably more than anything else. These are larger and they can eat more. And we have some that are wandering versus fixed that we might get into here in just a second. Eosinophils, somewhat phagocytic, they release toxins against parasites. So here's an image of what some of these look like. So for those macrophages, again, we see here, they're really just monocytes that have matured. And if you look at them, they look very similar. And the wandering versus fixed, if it's a wandering macrophage, then it can wander away. So it can leave the blood. It can actually move to the infection site. If it's a fixed macrophage, then it's going to be located in the liver, spleen, lymph nodes, the lungs, or the bone marrow, and it stays fixed into those positions. So for phagocytosis, what that process looks like is it's going to start with what we call chemotaxis. And chemotaxis is attraction. So it's the phagocytes' attraction to the microbes by following the chemicals. So chemotaxis. Once it gets there, it has to adhere. So this would be attachment of the phagocytes' plasma membrane to the microbe. And then it will ingest it and then digest it. So some organisms like Streptococcus pneumoniae, they have these capsules that inhibit adherence, which makes it very difficult for the phagocyte to actually bind to adhere to the microorganism. So complement proteins coat these microbes to promote adherence, and that can make it possible for them to actually phagocytize these microbes, and they call that coating process opsonization. And there's what that looks like. So we see that adherence, and then engulfing, and then digestion. So when an injury occurs, we've all been pricked by something, maybe a splinter. I know my brother one time got a treated it was a treated like 2x6, and he got a huge splinter in his finger, and it swelled up about 3 times the size of what the finger would normally be. So it had all of this inflammation, and you get that swelling, that redness. It'll feel painful. It'll feel warm to the touch. So all of these are signs of inflammation, and inflammation is defined as a local, non-specific, right? It doesn't matter what it was. It's not specific to what's happened. It's non-specific, and it's a defensive response. And all of these factors are actually helpful. Too much of it can be a bad thing, but there's a reason that you have inflammation. Now, chronic inflammation is a really bad thing because it damages your body. Your body's having to make concessions to have this type of inflammation, and in the same area over a long period of time can actually damage your body long term. So the four signs of inflammation, that's going to be redness, pain, heat. So we'll see a temperature increase, and that will be important for the porosity of the vascular tissue in that area. And then we'll see swelling. And all of these are actually useful signs of inflammation that actually help us fight off an infection. So the beneficial function of inflammation. Number one, it's going to destroy the damaging agents and help remove them. Number two, it can limit the effects of the agent by confining it to one place. And then number three, it repairs and replaces the damaged tissue. So inflammation is going to help facilitate these factors. So the three stages of inflammation would be, first, there's going to be vasodilation. So increased permeability, and that's good because it allows that vascular tissue to become more permeable, so white blood cells, phagocytes can move into the area, which would be the second stage. And then the last thing that will happen is, once the microbes are killed, is we need to repair the tissue that's been damaged by them, or the object that's been lodged in there, which is probably covered in microbes. So the third stage would be tissue repair. So that's what we see here. So inflammation in that early phase, we have neutrophil arrival. We said they were the first, or pioneering WBCs. And then our late phase, we have the monocytes, or the macrophages. So we have the proliferation phase, and then we have the remodeling phase. Some of these inflammation, or these inflammation pathways in general, are just pretty complex. And we're going to look at some of this at the end, and I'll show you some simplified diagrams to kind of picture what's going on. But at first, you have your innate immunity, and that's going to be for the first 4 hours of the invasion by the pathogen. And then it's going to move into this inflammatory response for the next several days. So what happens when we get a fever? So a fever, again, would be nonspecific. This would be a rise in temperature. And when it's systemic, it's going to cause your whole body temperature to increase. So usually you're going to measure that. You can measure that in the mouth. With infants, a lot of times they'll do a rectal thermometer, which is actually more accurate than the armpit. So it slows down bacterial reproduction rate. So that's obviously going to be super beneficial. And it increases the activity of phagocytes. So we decrease how fast they're reproducing, and we try to destroy the bacteria at the same time, and fevers help that happen. So it increases the production of interferon, and it's caused when phagocytes release interleukin-1. And interferon, that's a protein that interferes with the ability of a virus to reproduce. So, of course, with a viral infection, that could be really helpful. So, some other things, it can raise the body temperature, which is going to lead to that blood vessel constriction, increased rate of metabolism, and shivering. So the skin feels cold, which is going to produce those chills. When the body reaches a new temperature setting from the hypothalamus, the chills go away. So it creates those temporary chills. The higher temp will be maintained until interleukin-1 is gone. Then the hypothalamus will finally reset to normal body temperature. Heat-losing mechanisms are activated, vasodilation, and sweating. So they call that the crisis phase. So during a fever, the skin can feel cold because the body's temperature regulation system, which is the hypothalamus, what controls that, it's reset to a higher point now. So that means the body temporarily perceives itself to be below the new temperature setting because the hypothalamus has raised it for this inflammatory response. So then that's going to trigger these heat generating mechanisms, like you're going to shiver, you're going to have peripheral vasoconstriction, so narrowing of the blood vessels in the extremities, and that's going to try to raise your core temperature back up. So now that brings us to the specific defenses of the immune system. So just some general information here. We'd say immunity is a specific defensive response to an invasion by a foreign substance. So immunity targets specific pathogens, and we'll look at how it does this. So this is going to occur simultaneously with our non-specific responses. So we have those non-specific responses, and while that's happening, our immunity is also triggered to start specifically targeting pathogens rather than just these broad general mechanisms. So substances that provoke an immune response, we call those antigens. So to remind you the difference between an antigen and an antibody, we have a little microbe here covered in some surface proteins, and then we have an antibody. So the antigen is what is invading. And those are either going to be, proteins, large polysaccharides, some kind of component of the microbe that doesn't look like our own bodies. So it's something unique, something foreign. So then an antibody, which is really just a protein that we make, they're made to target the antigens. So antibodies are proteins that recognize and bind to specific antigens. So antibodies are going to help us neutralize and destroy these antigens. And you'll notice the way it's bound to it, the binding site complements the shape, the physical shape of the antigen. So those have to complement each other or the antibody can't bind to it. So it has to be specific to that antigen. Once you've been infected with something once and your body's already generated antibodies for that antigen, well, one, you can test for that antibody to see if you have been infected by the antigen in the past. And also, the second time you're infected, you'll already have antibodies that can readily be used to target that antigen for destruction. So that's when we talk about learning a pathogen, that's usually what we're talking about. So before birth, our lymphocytes actually are going to survey the body to figure out what is part of our body and what is not part of our body. So that way, when we are infected with something, our body can recognize whether it's self or non-self. So unspecialized cells form in adult bone marrow and fetal liver tissue. Some migrate to the bone marrow, we call those B cells, and some migrate to the thymus, we call those T cells, for bone cells and thymus cells. So then our B cells and T cells are distributed to the lymph nodes, the tonsils, the spleen, and the blood to monitor the body for foreign materials. So something that's pretty amazing about this is our bodies make 100 million lymphocytes every single day, which means we also have to destroy about 100 million lymphocytes every single day to keep that number in balance so they don't get too high. So again, we said these B cells and T cells, they're monitoring the body. When they encounter a foreign antigen, the B cells produce antibodies that bind to the antigen that then mark it for destruction. While T cells, they either directly kill the infected cells or they help coordinate the overall immune response. And we'll look at here in just a minute what that looks like. So our types of immunity, we have innate, which is non-specific, and then we have adaptive, which is going to be specific. The adaptive is going to be a little bit slower because it has to specifically and physically be able to connect to that specific antigen, not just having some general type of cell or system that destroys any pathogen. So 2, we have that actively acquired immunity, and then we also 3, we have artificially acquired immunity. So the innate, that's the resistance to those diseases that you're born with. It's non-specific. Actively acquired immunity, that would be that the person's immune system is stimulated by an antigen to produce an immune response. And then artificially acquired immunity would be the person is purposefully injected with an antigen, and then that allows them to build up immunization for it. So it was artificial because you've received that antigen artificially, not naturally. So for artificially acquired immunity, we get this injection. The inactivated bacterial toxins or toxoids can be given, such as tetanus, for example. You can get killed microorganisms. So they're already destroyed, but your body starts to develop antibodies specific to these parts of the microorganism, that specific microorganism. So if you're ever actually infected by it, your body's already trained to fight it off. You could get living but weakened or attenuated microbes, and those can occasionally produce the disease, actually, which is what we saw in the oral polio vaccine that we discussed previously. You could be exposed to parts of the microbes, such as just their capsules, and a new pertussis vaccine actually does that for us. So anyway, these usually can no longer cause disease, but do elicit an immune response. And again, another spell check issue here. For passively acquired immunity, for this, a person receives antibodies from another organism. So this could be naturally acquired passive immunity. For example, the antibodies that a mother has can be passed to her fetus through the placenta, or it can be artificially acquired passive immunity. So that's transferred from an injection of the antibodies from an organism that is already immune. So some interesting applications of this. You've probably heard of monoclonal antibodies during the whole COVID outbreak. That was something that was being used. And those are actually made in a lab and given to somebody. And so it's unlike a naturally produced antibody. These are made in a lab and they actually mimic the body's immune response and they bind to that specific target and then trigger the neutralization of that pathogen and the destruction of it. So for cancer treatments, they've talked about using those to bind to cancer cells, and then that marks them for destruction by the immune system, or it can block their growth signals. So that's something really interesting that's kind of cutting edge right now. So just to kind of summarize what this looks like. So for our active immunity, we have natural and artificial, and then for our passive immunity, we also have natural and artificial. So for active immunity, we have natural and artificial. So active immunity is when the body actively produces its own antibodies. So usually that's exposure to an antigen. So you get some kind of infection and your body produces its own antibodies to target it for that present infection and then for future infections. So usually you're going to get that naturally, but you can also get that artificially through some type of vaccination. So if we're talking about passive immunity, right, passive is when antibodies are transferred to the body from another source. So if that's natural, well, that could be from nursing, it's passed from the mother to the offspring, or it could be from, like we just mentioned, monoclonal antibodies made in a lab and then given to a person to provide some immediate protection, but that's generally going to be temporary. So we'll talk about immunity. And COVID's a good example of this, or really any type of virus or infection. You've probably heard of herd immunity before. This is where immunized people do not act as carriers. So if they have the immunization, then they're not going to pass it from one person to the next. So we talk about immunization campaigns, like everyone trying to get a particular vaccine. What it does, you'll see on the right, it says herd immunity achieved, and you have all the green. These are people who are immune. Maybe let's say they've received the vaccine or they've been infected by the disease in the past and they're immune to it. And then you have the infected person who can spread it. The little clearer people, they are susceptible to the virus or pathogen. As long as the herd or most of the people are immunized, then it's unlikely that two susceptible people, that one will be infected and then spread it to another susceptible person, because there's so many immune people in between the susceptible people. And they always do these calculations to figure out what percentage do you need to reach before you experience herd immunity. And that's why vaccination campaigns try to get as many people to get the vaccine as possible, because you see here another example, these blue dots, you increase the number of immune people so that the susceptible people don't come into contact with each other. So for example, with COVID, elderly people, people who are going through chemotherapy, like my brother at the time, they really needed not to be exposed to the virus from people who were susceptible and people who were infected and spreading it. So the more people you had vaccinated between vulnerable people, susceptible people, and some people, they can't get vaccines because they're under some intense type of chemotherapy and treatments. So if you have everyone else vaccinated, then it's less likely that those diseases are going to reach the elderly and the sick and those who are going through chemotherapy. For antibody-mediated immunity, or what we call humoral immunity, this would involve the production of antibodies. So they're going to be made by these B cells, and there's millions of these, right? We make 100 million different types of lymphocytes a day. So the immune response is triggered if an antigen enters the body and the B cell receptor has a close fit. It has to actually be able to connect. So we've got 100 million opportunities for this, and our body's constantly making new ones, and some of those will likely be able to connect to that antigen. So the two types of cells that are produced by B cells, one of those is called a plasma cell. We see this differentiation of the B cell here to a plasma cell and then a memory cell. So the plasma cells, they're actually going to make and release these antibodies that can target the antigens for destruction. Now, memory cells are also made. So these remain inactive in the body so that next time you're exposed to that same antigen, the memory cell will be able to actually initiate that immune response. So here we see a diagram of this primary and secondary response. What happens the first time we're infected versus the second time that we're infected. So we look at that first exposure, the first infection. You see you have that naive B cell, and then we have activated B cells, and then we're going to get our antibodies. They're going to be produced to target that antigen for destruction. And once the infection is taken care of, then we're going to decrease the antibody production. It's going to go back down. But we're going to have some memory B cells that are left over so that the second time we're infected, we can initiate that immune response faster. We already have the antibodies ready to go to target that pathogen for destruction. And if you look at how many days it takes to reach that peak response, the first exposure, 7 days. The second exposure, 3 days. And that response, the amount of antibody is much higher, which means we're going to have a much stronger and quick effect at actually destroying that antigen. And then it goes back down, and you'll look at after the second exposure, we have even more memory B cells. So we build up immunity after these first and second exposures, and we increase the amount of memory B cells that we make, which means we can make more antibodies and make them faster the next time we're infected. So 5 possible results of what happens with the antigen binding to the antibody. So the first thing, or one of the things that can happen, is neutralization. So that covers the harmful adhering sites of the microbe. We see that with the immunoglobulin A in our saliva. And we also see agglutination. So this causes the antigens to clump together, which enhances our ability to phagocytize them because we can eat a bunch at once rather than having to find them all separately. Opsonization, the antibody coats the antigen, which also enhances phagocytosis. Cytotoxicity, antibodies attached to the antigens of a cell mark the cell for destruction by nonspecific immune cells. And then complement activation. This activates inflammation in a complement protein, which punctures holes in the microbe surface, which can cause them to lyse. So we see both an immediate effect of the antibodies on the antigens and then a prolonged effect of enhancing the ability for other cells to come and actually destroy or phagocytize the microbes. So it can lyse them, it can neutralize them, clump them together, and also make them easier to be destroyed by other cells in the immune system. So what does an antibody actually look like if we zoom into it? So it's just made of a protein, right? We make this, it's made of protein. Another name for it is an immunoglobulin. So when you see immunoglobulin A, immunoglobulin G, these are just, that's the name for an antibody, and they're proteins. They're made up of 4 polypeptide chains. 2 are long, heavy chains. 2 are short, light chains. So you see here, we have these heavy chains, and then you'll see this term variable and constant. So variable means it can change between the different antibodies. Constant is going to stay the same. You see the constant heavy chain, excuse me, on the inside, and then the light chain on the outside. But the ends are always variable. And that's what actually changes to allow it to bind to different antigens, is going to be that variable portion of the immunoglobulin. So the different classes of immunoglobulins, and again, that's just another term for antibodies. You can remember the acronym GAMED, and we'll show you some ways to remember that. This is not in that exact order, but GMADE. So for IgD, this is the most abundant in serum, only ones that can cross the placenta and protect against circulating bacteria and viruses. They neutralize toxins, trigger the complement system, and enhance phagocytosis. Immunoglobulin M, these are the largest antibodies. They are the first to appear in an immune response after to the initial exposure, and the presence of them indicates a recent infection. Immunoglobulin A, that's what we saw in our tears and in our saliva, and that's actually the most abundant in the body. And it protects our mucous membranes as well. IgD, found in the blood, lymph, and on the surface of B cells, and then IgE participates in allergic reactions. So here's how you can remember the GAMED antibody classes. So the first one, for the G, this is the only one that can cross the placenta during gestation. So G for gestation. A, immunoglobulin A, this is going to be the nose and other airway passages or places with mucous membranes. M, this would be immediately, are going to show up first to fight an infection. So M for immediately. E is what we see during allergic responses. So allergies, E for allergy, and then D, cause B cell differentiation, D for differentiation. This little image here is a summary of our humoral immunity versus cell-mediated immunity. And there's a video by a guy named Immunology. If you look up humoral versus cell-mediated immunity, he goes through this nice, clear drawing to kind of show you what's going on. But just to summarize briefly, but I would recommend watching that video. It's Immunology, humoral immunity versus cell-mediated immunity. But anyway, for B cells, you'll notice it's summarized as antibody-mediated because we're eventually going to get antibodies from this pathway. And that's going to neutralize and target the pathogen for destruction. For our cell-mediated, this controls intracellular pathogens. So cell-mediated means in the cell. So our T cells are going to play a major role in this. And eventually what's going to happen is the antigens are presented on the surface of an infected cell. And then T cells are going to target them. It's going to trigger a pathway that's eventually going to lead to their destruction. So I definitely recommend watching that video. I hope this video helped you learn more about our first, second, third lines of defenses and immunity.